Okay, let us begin. In the Father, Son, Holy Spirit. Amen. God, grant me serenity to accept the things I cannot change, courage to change the things I can, wisdom to know the difference. Father, Son, Holy Spirit. Okay, let's see here. Got Jasmine, no. I think I know where she is. Callum. I got Aiden. That's probably okay. Emily. Hey, Emily. Abigail. Ashley. Anna. I saw Anna. She's here. She stopped in, right? Didn't she pop in right there? And she disappeared? No? Okay. Got John Hurley. Quattro. Okay. Cameron in the back. Rebecca. Okay. John Cost. Kate. There's Callum. Sophia. Marie. Sam. Yo. Yeah, that's online. Christina. Kristen. There's Kristen. Hey, Kristen. That's Sarah. Tristan. Jordan right there. Antonia. Deshaun. Saw you come in. I know Joan is here. Landon. No, we got Anthony in the back. All right. Becca. Okay. Caleb, Matthew, Kyle, Olivia. All right. So let's rock and roll. I'm not gonna be able to move this now. Nope. So we're gonna cover 4 processes. These 4. For each one, man, you need to know the beginning molecule, what you start with, what you end with, what you get, and where the process happens. We're going to see that where this process happens is going to be different depending on whether it's a prokaryotic cell or a eukaryotic cell. Okay? And we're going to review glycolysis real briefly here in a moment. So, we said that glycolysis literally means killing glucose. So it beginning molecule is a 6-carbon glucose. Boom. You go through that process, end product, 2 3-carbon pyruvates. So we still have all of our carbons so far, right? They're all accounted for. We'll talk about other products in a moment. So what do we do with the pyruvate? There's a couple of things that we can do. One, is we put it through fermentation. So in yeast cells, the end product of this fermentation process is ethanol. So we go from a 3-carbon pyruvate to a 2-carbon ethanol, and we lose a carbon dioxide in the process. And this step right here, where we oxidize NADH back to NAD, that's really important. Now in animal cells, any animals in the room today? Animals? Any animals here? We got a few animals. You animal, you're just a bunch of animals. No, we're better, we're different than that. But anyways, yes, we are animals. So in our cells, the pyruvate, if it's undergoing anaerobic respiration, if the cells are utilizing anaerobic respiratory processes, the pyruvate is converted to lactate. No carbon dioxide as a byproduct, but we notice that the same process is happening here. NADH gets oxidized back to NAD. Why is that important? If we don't reclaim, if we don't regenerate our NADs, glycolysis is going to get shut down. I need NAD to carry out the full glycolysis cycle. So if all of my NADs are converted to NADH in the cells, this process is going to go stop. It's going to hit a wall, and you're going to hit a wall. So you can't, anyways. So this is a major reason why this process is very important. Helps keep glycolysis going forward. By the way, where does glycolysis happen in cells? Kristen, where does glycolysis happen in cells? Cytosol. Look, I'm going to ask some review questions today. I know that you guys, a bunch of you have your lab exam today, so you're really not going to be here. So that's okay. That just means I'm really not going to like you. I'm just joking. Okay, so we'll move ahead. So remember, we start with the glucose, 6 carbons. We're priming that with 2 ATPs. And we need, what else do we need? We need 2 NAD pluses. So we need all 3 of these things to make this process go forward. Okay, and I've already said, oh, what form is NADH? Is this the oxidized or the reduced form? Oxidized or reduced form? It's reduced. Now I am on Ferris Bueller. Anyone? It's RE. Anyone? Reduced, right? Yes. So NADH is the reduced form. Okay. Sweet. So big part of this, replenish NAD. We need that. All right. So, glycolysis happens in the cytosol. Start with a 6-carbon glucose. We get 2 3-carbon pyruvates. What do we do with that next? If it's anaerobic, if it's in a yeast cell, it goes through fermentation. It's still kind of fermentation in animal, and the end product is ethanol. If it's in an animal cell, the end product is lactate. Well, what if we're not doing anaerobic oxidation or respiration? What if we're undergoing aerobic respiration? Well, the cell takes that pyruvate and now it's going to take it through a different process called pyruvate oxidation. Pyruvate oxidation. Think of the process. What's that word mean? I'm taking it from a higher energy state to a lower energy state, right? Is that right? Yes. So we start with pyruvate, and then here's another player here. We're going to end up with acetyl-CoA. Here's my beginning, here's my ending. And what happens is, and by the way, how many carbons in pyruvate? How many carbons in pyruvate? Tristan, how many carbons in pyruvate? How many carbons in acetyl-CoA? 2. Dun, dun, dun. Something's going to happen to a carbon. I bet we can predict what's going to happen, but we'll see in a moment. So pyruvate is added to coenzyme A. That just means coenzyme A. The end product of this process of pyruvate oxidation is acetyl-CoA and some byproducts. So the end product is acetyl-CoA, and we also get NADH. So we're going to get the reduction of NAD. And then what do you think happens to that carbon, Christina? What happens to that carbon? Where does it go? Where do we go? Where do we go now? Where do we go? What does this even look like? What is that? That actually looks like something that we're familiar with, right? Yo! So we go 3, doing, we lose 1, yes. So, carbon dioxide. So we lose, there's a carbon dioxide as a byproduct. So pyruvate undergoes pyruvate oxidation. The ending molecule is acetyl-CoA, and our products are NADH and carbon dioxide. Good. So keep that in mind. Anytime we lose a carbon, it's going to be lost as carbon dioxide. So let's count it up. Ching. You guys don't even know what that is. Ching. Do you know what that is? That's what I thought. It's an old adding machine. Ching. Anyways, so how many pyruvate molecules, let's stop for a moment, are produced per molecule of glucose in glycolysis? How many pyruvates do we get? 2, right? 6 carbon glucose, 2, 3 carbon pyruvates. Boom. So, in this step, how many more NADHs did I get per molecule of glucose? This is Anthony. Why'd you say that? Okay. I get 2 more NADHs per glucose. 1 glucose, 2 pyruvates. Each process of pyruvate oxidation produces an additional NADH, so I get 2 more NADHs. Are we cool with that? So, Net total so far per molecule of glucose. ATP, 2. We get that from glycolysis. Right, net. We put 2 in, you put 2 in, and you get 4 out. You put da-da-da, you get 2 ATPs, right? So we end up with 2 ATPs net from glycolysis. How many NADHs have we produced so far? Marie, you were looking. How many? 4. 2 from glycolysis, 2 from pyruvate oxidation. Sweet. And so far, we've also produced now 2 molecules of CO2. So pyruvate oxidation, we lose CO2. Keep that in mind. 2. So at this point, we've started with 6 carbon glucose. Now we're down to 2 carbon acetyl-CoAs. Cool. And we still haven't produced very much energy yet. So we gotta get rocking. But where does pyruvate oxidation happen? In eukaryotes, it happens in your mitochondria. Hey, crikey. Look at that. Mitochondria. It happens in the mitochondria. Well, but first we got to transport it in, and we're going to keep that in mind. Well, why would this thing have to be transported? Why would this thing have to be transported into the mitochondrial matrix? Why would it have to be transported to pass through a membrane? First of all, because pyruvate is polar. Thank you. I knew you knew that. Good. In prokaryotes, this happens in the cytosol. All right. Now it's time for the crazy ride. The Krebs cycle. The Krebs cycle. Citric acid cycle, TCA cycle. Has like 3,000 names. We're going to call it the Krebs cycle. First of all, it is a cycle. It's like a wheel. Isn't that lovely? That's so lovely. And what that means is that the beginning molecule, which is oxaloacetate, is the same as the ending molecule. Oxaloacetate, same molecule begins this cycle as the molecule that ends. So one of the faculty members in the biology department likes to likens this to like one of those teen horror movies. Teen horror movie, like The Haunted Ride. And the car comes up, tick, 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 pulls into the station. And there's probably like all kinds of, it looks nice and clean. And there's four teenagers in there. 1, 2, 3, 4, there they are. They're like, hey. And of course, they act like really strangely, right? But nobody ever notices these things in those horror movies, right? You never notice that. They're just like, hey, this is a great ride. Would you like to go on this ride? And the two that are in line, like, this sounds great. Let's get on. Hop, hop. So they drop on the car. So these two right here, these two little carbons, 1, 2, donated by acetyl-CoA. So this is the role of acetyl-CoA. So once acetyl-CoA is formed, then it donates its 2 carbons to oxaloacetate. Maybe they'd be like, I don't think this is a good idea. Do you really want to go on this? Yeah, that sounds like a really good ride. We're going to get on. So they get on. And they get in. So now we have this 6-carbon molecule. And then it goes through the cycle. And then basically at different points in the cycle, these two unsuspecting carbons, which jumped on this nice little fun ride, they get thrown out of the car. Wah! Here goes one. Yo! Here goes the other one. Ah! That's carbon dioxide. And then it comes back to the beginning. Click. Hey, this is a really fun ride. Do you want to get on? Weren't you the same 4 kids that were, anyways? So, yes, yes, yes. Now, the molecule that is actually formed here is called citrate. Citrate. Which is why this is then called the citric acid cycle. That's why one of its other names is citric acid cycle. So the big point here is that we start with a 4-carbon molecule. Acetyl-CoA donates 2 carbons to this 4-carbon molecule. If you want to know it's oxaloacetate, great. And we then get a 6-carbon molecule called citrate. Then it goes through the series of reactions. And what are all the products that we see here? Name them all. Take a look. Let's get them down here. What all do we get? What all do we get? It's called the soft shoe. Never took a single lesson. So what are some of the products? Sarah, what are some of the products? Give me one. How many? Well, we're gonna let's keep it to one cycle here. So how many per cycle? There's an ATP right there. I don't even know if I have that. Okay, so let's look at per molecule of acetyl-CoA, then we'll do the higher math of multiplying everything by 2 later. So, okay, we get ATP. What else do we get from here? Is it Ashley? Ashley. How many? Okay, that's actually not an NADH. So we've got 1, 2, 3. Okay, that's hard to see. That's hard to see. Okay, NADH. 1, 2, 3. Sweet. 3. What else do we get? So we have ATP, NADH. What else do we get? Yep, Deshaun. Carbon dioxides. Good. We get carbon dioxides. And then we've got this thing up here. This is actually FADH2. It's hard to see. Oh, FADH2. Boom. Right there. We get one of those. ATP. ATP. It's my energy. It has phosphates 3. Hey, we need water too. Good. 1. Finally, carbon dioxides. 1, 2. Good. 2 of those. So, I ask you, once these carbon dioxides are flung off, see, these are flung out of the car. Gone. Woo! Gone. You know, nobody survives this trip. All the carbons are thrown off as carbon dioxide. So, if we had 2 acetyl-CoAs, so our initial glucose, 6 carbons, We're down to 2 3-carbon pyruvates. We're then down to 2 2-carbon acetyl-CoAs. We've already lost 2 of our carbons as carbon dioxide, right? Now, we've lost the rest of them. So by the time the Krebs cycle is finished, all of our carbons from glucose are bye-bye. Bye-bye, Miss American Pie. Yeah, gone. So, they're lost as CO2. So all the carbons are gone. So, let's add up our molecules. Now we're going to go back. Sarah, now we'll go back and we will tally all of our molecules that we've produced so far per molecule of glucose. So not to confuse the matter, but here we looked at per molecule of acetyl-CoA. And when I want to calculate how many molecules of each are produced per glucose, what do I have to do? I have to multiply all these numbers by 2, right? Because I get 2 acetyl-CoAs per glucose. All right, let's do this. How many ATPs do we have so far? Quattro. 2? We got 2 from here. Did we get 2 from somewhere else? You sure? How about glycolysis? Did we get any from glycolysis? How many did we get from glycolysis? 2. Okay, that's all right. I know you guys got lab exam. I'm sorry I'm like distracting you from trying to remember what you want to remember for your exam today. 2 from glycolysis, 2 from the Krebs cycle. 2 from glycolysis, 2 from the Krebs cycle. How many NADHs have we done? You can look over your notes and take a look at it. We have generated a lot of NADHs so far. I'm going to tell you that it's 10. 10. 2 from glycolysis. 2 from pyruvate oxidation. 6, right? 6 from the Krebs cycle because I get 3 per molecule of acetyl-CoA. 2, 2, and 6. Last time I checked, that equals 10. But you know, if you do some forms of math, 1 plus 1 doesn't equal 2. But don't go there. That's like calculus that we don't like because you use weird numbers. Anyways, and then we get FADH2. We get 2 of those from the Krebs cycle. And we have produced 6 molecules of carbon dioxide, right? 2 from pyruvate oxidation and 4 from the Krebs cycle. All right. These are just numbers. So we've gone all the way, all of our carbons are gone, and we still only produced 4 molecules of ATP. This doesn't seem like a very good system. So what are we going to do now? Well, those of you who've studied this before, oh shoot. Where does this happen? Eukaryotes, the matrix. It's in the mitochondrial matrix. And in prokaryotes, it happens in the cytosol. Cytosol. Does whatever a cytosol does. Do I have my dry erase marker? I do not, but I have a mask. Yep. So we've gone through a lot of work so far. We still don't have much ATP. So the final step is through this chain, which is called the electron transport chain. Electron transport chain. We're going to be transferring electrons from molecules, from protein to protein. So what are going to be the big players here? NADH and FADH2. Why? What's their role in the cell? They are electron carriers, right? So that NADH, that's the reduced form. FADH2, that's the reduced form. So these molecules are jazzed and ready to go. It's like you, after 3, does people even drink Red Bull anymore or anything like that? You just drink coffee. You just say, forget the fancy stuff, I just want my coffee. All right. Okay, electron transport chain. Okay, let's see here. I just want to do a sound check. So you guys still hearing me out there? You still hearing me? Check, check.
Yeah, I can hear you.
Okay, get ready. We're going to go crazy in the classroom here, so hold on. All right. It's electron transport chain. You look worried. Are you worried? You should be. So, what is it? First of all, what's this thing? What's this thing? John Coast. Phospholipids. So we got a bilayer, right? Phospholipid bilayer. Sweet. What are these representing? These are proteins, transmembrane proteins. So the electron transport chain, it's literally, just think of it as a series of proteins. These are all representing these are enzymes. Q and C, these are enzymes. But enzymes are also proteins. They're all in a chain. So here's what happens. Whoa! What the heck? So as we step into the mitochondria has 2 layers of membranes. We got the outer layer, and then we have the inner layer, and then inside here, inside the second layer is the matrix, the mitochondrial matrix, not the red pill, whatever. You guys don't know what that means. Okay, so, and then here we have the intermembrane space. So this is the outside layer of the mitochondria right here. This is the outside layer. Okay, so here's what happens. NADH, man, it's got the electrons. And it comes, and it hands it off to the first protein. Now it's got this energy. Whoa, dude! It's fired up. It's got these electrons. And they're moving, they're grooving, they're shaking and baking, right? So NADH donates 2 electrons to the first protein here. And so then NADH gets oxidized back to NAD. Goes back to being calm. But then what happens? Oh, the protein has got this energy. It's not just a protein. It is a H+, that's a proton, hydrogen ion. So it's also a transporter. It moves hydrogen ions from the matrix out here to the intermembrane space. It transports it. It actively transports it. And it transports multiple protons. All right? Moves them. Then the electron gets passed on down the line. So as it gets passed down the line, jung jung, keeps moving. It hits these different proteins like NADH dehydrogenase, right? It tells you what it does. Dehydrogenase. BC1 complex, this protein, it receives a protein or an electron, and it moves hydrogen ions from one side, from the matrix out to the intermembrane space. This electron then gets moved to C, this enzyme C, and then it gets to this one, cytochrome oxidase complex. Keeps moving these protons. So these electrons, which were initially donated by NADH to this NADH dehydrogenase, it just gets bounced. It'd be like if we were all in a relay chain. Got it, got it, got it. Oh, and I got to move this too. So it's like one of these things. And all this is to move all of these hydrogen ions into this intermembrane space. And then finally, what happens to the electron when it gets to the end of the line? It combines with oxygen. Oxygen is the final electron receiver of the electrons. It's a big role for oxygen here. Notice this is the first time we've even talked about oxygen, right? We said this whole process is aerobic respiration, which requires oxygen. This is the only time we've even mentioned oxygen. So it's the final acceptor of electrons. That's its role. Without oxygen, this whole process would shut down. Because all these steps would back up. Because what you do is unless this protein here can donate the electrons to oxygen, then the whole chain is going to stop. The whole assembly line stops. Okay. So let's stop there for a second. Any confusion, any questions about this process in general? John?
So the hydrogens are coming across the membrane protein because the electrons are carrying them across the charge.
So the electrons, okay, it's a good question. The electrons give the protein the energy that they need to move the hydrogen ions. Okay, it's a good way, it's a good question. And we're going to see why moving all of these hydrogen ions is really important. Because what's going to happen is the cell is going to use all those hydrogen ions, let them pass back through the plasma membrane, and that's how we make ATP. Now, we talked about NADH. What about FADH2? It pops in right here at this coenzyme Q, this little boop. So FADH2, it donates its electrons to this enzyme. And then those enzymes then go through the other two proteins. All right. So as we moved all of these protons from the matrix out to this intermembrane space, now we're going to get a different look at this, okay? So here, we're reorienting ourselves. This is the matrix up here. This is the matrix up here. This is that inner mitochondrial membrane. This is the outer mitochondrial membrane. And this is the intermembrane space. So here's our three transport proteins, right? Moving hydrogen ions from the matrix out to this space. And as these hydrogen ions are transported, the concentration of hydrogen ions is increasing here. So the concentration of hydrogen ions in this space, this intermembrane space, is very high. And in the matrix, it's very low. Okay, so now what do I have? I now have I've got a lot of hydrogen ions in the intermembrane space. I don't have very many at all inside the mitochondrial membrane, or in the matrix, excuse me. So now there's that concentration gradient for hydrogen ions to move. If there's a lot here and not very much here, that means that there's a concentration gradient for hydrogen ions to move from the intermembrane space where? Back into the matrix. So there's a concentration gradient. All those hydrogen ions have been moved, using that energy, the energy of the electrons. But I ask you, can hydrogen ions easily pass through the plasma membrane? Give me a yes. Give me a no. Give me an I don't know. Okay, we got some folks who are like, I don't know. Okay, good. Thanks for the honesty.
No.
Oh, what was that?
No.
Okay, no. I've got a no from the internet. A no from the internet. Charged molecules. What are they going to hit? They're going to hit the non-polar tails of this inner mitochondrial membrane. Remember that. They're going to hit that non-polar region, and they will be stopped. So the hydrogen ions cannot easily pass back into the matrix. Just like they needed help moving from the matrix out to the intermembrane space, right? They needed these transport proteins to move them. Okay? So now we need a carrier protein to let them move back through the inner mitochondrial membrane. Okay, so now I have a concentration gradient, which is a form of potential energy for the hydrogen ions to move back into the matrix. That means when it does move, that potential energy gets converted into what form of energy? Kinetic energy, right? So once they start moving, your cell's going to use that to do some work. Right, they do. They move back in through a protein, big protein called ATP synthase. I bet we know what this does. It makes ATP. It tells us. The name of the protein tells us what it does. It makes ATP. And this is it. This thing is monstrous. It's a mega protein. It's actually a rotor, which is pretty cool. So it's a complex pore for hydrogen ions. We good on time? Yes, we are. So here's our hydrogen ions. They're high in the intermembrane space. They're low in the matrix. And these things start passing through this protein. Zhoo, 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 zhoo. And this thing literally spins. Just blew up my pointer. It literally spins. Scientists have actually taken a little magnesium rod, teeny, itsy, little, small. You need a really low resolving power to see this thing. They've attached it to ATP synthase, and they've watched it spin. It's crazy. So what happens, this thing moves through, this thing begins to move. And the ATP synthase turns. And it produces mechanical energy that's then basically used by the ATP synthase to convert ADP to ATP. So the real simple story. The electron transport chain uses the energy from the electrons to move hydrogen ions from the matrix to actively transport hydrogen ions from the mitochondrial matrix out to this intramembrane space. This creates a concentration gradient that would favor the movement, the passive movement of hydrogen ions back into the matrix. It does that through ATP synthase. So as the hydrogen ions move through, it spins, it moves this protein, and then ATP synthase uses that energy to convert ADP to ATP. It takes an inorganic phosphate, it forms a covalent bond through the process of dehydration synthesis using energy to form the bond. Maybe it's dehydration synthesis. It might not be. But anyways, we're forming a bond there. We're using energy to form the bond. Boom. Whoa! And that's the end. So, on Wednesday, we are finishing this section. We are going to basically go through all four processes. We're going to summarize all of our molecules. We're going to see how many of what we get from what, where things happen, et cetera, et cetera. And then we'll be ready for Friday. It's actually only 10:19. Vamos! Is that a word? Vamos? Is that vamos? Andiamo. Allons-y. Au revoir. Que sera, sera. Hasta la vista, baby.